Hello, my name is Kevin Korn, and I'm a third-year PhD student in Marjan Rafat's lab at Vanderbilt University. And today I'll be talking about my project titled Understanding the Roles of Lean and Obese Adipose Tissue on Triple-Negative Breast Cancer Recurrence. Breast cancer was the leading cause of cancer-related deaths for women worldwide in 2018, with over 2 million new cases and 600,000 deaths attributed to the disease. This year in the United States, it's estimated that breast cancer will be the second leading cause of cancer-related deaths for women, with over 250,000 new cases. Now, our lab is especially interested in triple-negative breast cancer, which has a poor prognosis and is a highly aggressive and metastatic subtype. It represents about 15% of all diagnoses and generally has a poor prognosis with about a 15% lower survival rate compared to other subtypes. Additionally, triple-negative breast cancer disproportionately affects young African American and Hispanic women, as well as those with the BRCA1 mutation. Now, triple-negative breast cancer is called such because it lacks expression of the estrogen and progesterone receptors, as well as lacks the overexpression of the human epidermal growth factor receptor 2, which means that triple-negative breast cancer lacks targeted therapies that other breast cancer subtypes can be treated with. Now, since triple-negative breast cancer lacks these targeted therapies, treatment typically involves surgical intervention in the form of a lumpectomy or mastectomy, where the entire breast tissue is removed, adjuvant or neoadjuvant chemotherapy, as well as radiation therapy, typically given in a fractionated regime, which means about 2 gray doses are given once a day for 4 to 5 days a week across multiple weeks, leading up to about a total dose of around 50 gray. Now, even with this treatment modality, triple-negative breast cancer has a relatively high rate of local regional recurrence, regardless if a lumpectomy or mastectomy is performed. And this local regional recurrence typically comes about due to one of 2 ways. Cancer cells might be left behind at the primary site after treatment because they became resistant to treatment and then over time grow into a secondary tumor. Another method could be that circulating tumor cells shed off from the primary tumor during initial progression and then are recruited back to the primary site due to chemotactic cues from their damaged tissue. Now, this circulating tumor cell recurrence model was demonstrated in a preclinical model utilizing female BALB/c mice, where luciferase-labeled 4T1 triple-negative breast cancer cells were injected into the number 4 mammary fat pad, and then in the contralateral mammary fat pad, instead of injecting tumor cells, instead that fat pad was irradiated with a 20 gray dose of ionizing radiation, and then the triple-negative breast cancer cells were tracked over time and were shown to migrate towards that radiation-damaged mammary fat pad by day 10 post-radiation. And it was determined that the circulating tumor cell recruitment was preceded by a large flux of macrophages, which was abrogated in the presence of CD8 positive T cells, potentially indicating that the radiation-damaged normal tissue may recruit circulating tumor cells and ultimately facilitate triple-negative breast cancer recurrence in certain immune statuses. Therefore, we really wanted to better understand how the normal tissue responds to radiation damage impact recurrence, not just by evaluating what signals the damaged adipose tissue cells give off to recruit immune and cancer cells, but to see how radiation damage impacts the local microenvironment to provide an area that's favorable for tumor regrowth. And we wanted to focus in on adipocytes because not only are they a major player within adipose tissue, but when they become cancer associated, they can provide signaling to cancer cells through adipokines and also provide fuel for cancer cell growth through the release of free fatty acids. We are additionally interested in how obesity impacts all of these processes, since several studies have shown that obesity ultimately leads to increased breast cancer progression, where patients that are severely obese have a decreased probability of disease-free survival, as shown in this Kaplan-Meier curve. And then if we focus on invasive breast cancer, specifically in regards to local regional recurrence, patients that have an above median body mass index have a lower probability of survival once they develop local regional recurrence. So we are really interested in understanding how obesity impacts the breast tissue microenvironmental niche, how obesity alters the wound healing response in regards to radiation damage as part of radiation therapy, and then ultimately how obesity impacts both cancer cell recruitment and tumor regrowth. In order to begin answering some of these questions, we employed an in vivo diet-induced obesity mouse model, where we took female Balanced mice that were 3 weeks of age, and after an acclimation period, introduced them to either a low-fat or very high-fat diet for 15 weeks, where the very high-fat diet contained 60% kilocalories from fat, and the low-fat diet contained 10% kilocalories from fat and was sucrose matched to the high-fat diet. Across the 15 weeks, the high-fat diet group gained statistically significant more weight compared to the low-fat diet group. And we evaluated other obese characteristics such as serum cholesterol and triglyceride levels, in which case the high-fat diet group contained higher levels in comparison to the low-fat diet group. And finally, examining the livers of the high-fat diet group, we observed microsteatosis, which was not observed in the low-fat diet group. We took 2 approaches in evaluating the wound healing response in the lean and obese mice. In the first scenario, we utilized ex vivo radiation without the injection of any tumor cells to see how those resident tissue cells responded to radiation damage without the complicating factor of infiltrating immune and stromal cells. Secondly, we performed in vivo radiation, still without tumor cells, to see how the infiltrating immune and stromal cells change the microenvironment after radiation damage. So for our ex vivo protocol, we excised the number 4 mammary fat pads and irradiated them ex vivo to 0 or 20 gray, incubated them for 2 days in serum-free media, at which point we analyzed the tissues via microscopy and collected the conditioned media to perform a 31-plex Luminex assay. In order to begin examining the cytokine secretions from the lean and obese mammary fat pads, we began by comparing the lean irradiated and obese irradiated fat pads to the lean unirradiated controls, and examined the top hits from our Luminex analysis, which were CXCL2, VEGF, CXCL10, and CCL11. Now, CXCL2 it has been implicated in neutrophil recruitment, which when we consider the mammary fat pad microenvironment, where we expect there to be higher levels of free fatty acids, those fatty acids have been shown to corrupt neutrophils into suppressor cells. Additionally, CXCL10 generally leads to lymphocyte recruitment, and CCL11 recruits eosinophils and has been shown to increase the migration and invasion of cancer cells. So not only could some of these infiltrating immune cells in the obese case potentially promote a immune suppressive and tumor promoting microenvironment, but the obese mammary fat pads could exhibit a unique immune cell profile compared to the lean fat pads after radiation damage. Next, we wanted to explore what was going on within the microenvironment at this time point. And to begin answering that question, we started off by looking at ECM proteins through immunofluorescence staining of collagen 1 and collagen 4. Here showing representative images of the unirradiated lean and obese fat pads, where collagen 1 is stained green and the nuclei are in blue. And it started to become apparent to us that there was decreased staining for collagen in the obese mammary fat pads, which was confirmed through quantification of the images, where there was a decrease in integrated density, which is the product of intensity and the area of staining. Now, looking at collagen 1, there didn't appear to be that much of a difference in terms of trends with collagen 1 staining from the irradiated to unirradiated groups. However, when we looked at similar staining for collagen 4, the difference between lean and obese fat pads in the unirradiated groups became more apparent. And it was interesting to note that when looking at the lean fat pads going from 0 to 20 gray radiation dose, there was actually a decrease in collagen 4 staining, suggesting that without infiltrating immune and stromal cells, perhaps the adipocytes are involved in ECM degradation as a result of radiation damage. However, not having infiltrating immune and stromal cells isn't necessarily the most biologically relevant, so we moved next to in vivo radiation, where we irradiated the fat pads to either 0 or 20 gray dose, and then waited 10 days to allow for wound healing to occur, at which point we analyzed the fat pads via microscopy to also, again, look at collagen staining. Here showing representative images at this 10-day time point for the lean and obese groups with 0 and 20 gray radiation doses. Again, collagen 1 staining shown in green, and it became very apparent, again, that there was still, in the unirradiated case, decreased collagen 1 staining in the obese fat pads compared to lean fat pads. And as expected with infiltrating cells, there was ECM deposition in both the lean and obese cases after radiation damage, which was again confirmed via quantification through integrated density. But it just became very apparent from the quantification of both collagen 1 and collagen 4 that the obese mammary fat pads just do not have as much ECM deposition for collagen 1 and collagen 4 compared to the lean counterparts. Now, how this changes when looking at other collagen, like collagen 6, which is very important within adipose tissue, or other proteins like elastin, which has been commonly shown to be associated with adipose tissue macrophages, remains to be seen, and we are looking at that staining next to see if we see the same trends or if something is different with those proteins. Next, we wanted to switch gears to evaluate how specifically adipocytes respond to radiation damage and whether or not they impact triple-negative breast cancer recurrence. To do this, we utilized 3T3L1 adipocytes that were irradiated in vitro to either 0 or 10 gray. These cells were incubated for 2 or 7 days after radiation damage, at which point their conditioned media was collected. Next, we utilized luciferase-labeled 4T1 triple-negative breast cancer cells and exposed them to that conditioned media for 48 hours, at which point we evaluated their proliferation via bioluminescence imaging. We observed that breast cancer cells exposed to irradiated adipocyte conditioned media collected 7 days after radiation damage had a significant increase in proliferation when compared to breast cancer cells exposed to unirradiated adipocyte conditioned media. Now, the 7-day time point became increasingly interesting to us as we evaluated the metabolic state of adipocytes after radiation damage. Here, I'm showing the expression of GLUT4 in green, where the nuclei of adipocytes are in blue. GLUT4 is the insulin-sensitive glucose transporter, and we observed that at 7 days post-radiation, radiation-damaged adipocytes had higher expression of membrane-bound GLUT4 compared to unirradiated adipocytes, suggesting that the irradiated adipocytes 7 days after radiation damage have a potentially higher glycolytic capacity. And how this relates to their ability to increase proliferation of cancer cells remains to be seen, but suggests really interesting future work for us. In summary, I hope I've demonstrated that the obese microenvironment exhibits unique secretions in response to radiation damage, that collagen deposition after radiation within mammary fat pads is impacted by the lean or obese status of mice, and that adipocytes ultimately impact cancer cell proliferation after radiation damage. With that, I would like to thank my advisor, Dr. Marjan Rafat, and Dr. Alyssa Hasty, who at Vanderbilt University has been very instrumental in helping us establish our obese mouse model, as well as our funding sources. Finally, I would be happy to answer any questions about the work that I presented today. Please feel free to reach out to me during the AIChE conference, or you can email me at kevin.c.korn@vanderbilt.edu. And with that, thanks for watching.